We are going to start the second morning session. And the first talk is going to be from Jürgen Cox, who of course doesn't need introduction in the proteomics community, but because this is a more mixed audience, I'll say that he is the person behind the most popular software platform for analyzing shotgun data-dependent acquisition mass-spec data. It's open source platform, free to use for everybody. And not only that it provides identified and quantified peptides, but it provides lots of other features from the analysis of the mass spectra that we have found incredibly useful in our optimizational workflows. So I've always been so grateful and excited that Jürgen is interested in using his skills and talents to advance the analysis of single-cell proteomics data. And I'm looking forward to hearing what are the new features of MaxQuant that are going to help us extract more from the data that our instruments acquire.
Yeah, hello everybody. So indeed, thank you for the introduction. I'm very happy to be invited also. So indeed, I want to tell you a little bit about what we're currently working on in MaxQuant to support single-cell proteomics data. So this is actually very much work in progress, what I'm reporting on. So I'm actually very happy that together with Nikolai and Bogdan, we're able to work on this data that they produce and get instant feedback and to learn immediately what we do in MaxQuant, if it's helpful, going in the right direction or not. So I think that's actually a very good environment to develop MaxQuant further, to make it useful for the single-cell community. And so that's basically what we want to do. So we want to support the TMT-based single-cell proteomics workflow and provide improvements in MaxQuant, which is essentially improvement in the TMT workflow for MaxQuant, of course. And also, so we have this other software here called Perseus, provide all the tools that are needed here to do the downstream analysis for the single-cell proteomics data. So just in case you don't know MaxQuant and Perseus and what I'm talking about, so MaxQuant is basically the software that does the data analysis of the spectra. So it takes this mass spectrometric data as input and outputs the protein identifications and quantifications. And this then would normally be handed over to this Perseus software, which does then the downstream analysis, right? And so there are two improvements in MaxQuant that we're working on that are very relevant for a single-cell proteomics data. So one is the isobaric match between runs, as we call it. So I will explain later what that means. And the other one is a specific normalization for TMT data, which is on the PSM level, and so which is called PSM level weighted ratio normalization. Then what will be going into Perseus is basically all the basic stuff that you need to handle, like huge amounts of TMT batches, which you usually get in single-cell data. So some automated tools for annotating isobaric labeling data. I mean, just get the correct sample names, for example, attached to the different label channels and so on. That's actually, if you have now hundreds or something like this of batches, it's actually already a non-trivial task to do. So we provide tools for doing this in a streamlined fashion. Basic filtering of channels, get rid of carrier channels, of empty channels and things like this. Filtering of or detection, first of all, of low quality samples, and then also get rid of these for the further analysis. You want to do, or you want not to do imputation, and for that you want to have several methods available that you can compare against each other. Then, of course, removing batch effects on several levels. So we do this already with the normalization that we do here on the level of PSM normalization, but also then on top of that, you might want to correct for batch effects on the protein quantification level. So there would also be tools for doing this available here. Then, of course, the dimensionality reduction tools are a very important point in this, and then comparison of samples and, for example, doing differential expression of single cell data. So that's the program, and we're somewhere in the middle. So that's why, as I said, this is very much work in progress, what I'm reporting on, and the final software might actually look slightly different, and the final tools and algorithms might actually not be exactly what I'm telling you now. Actually, one point of this is actually getting feedback of what you want, actually, right? So. Okay, so yeah, the way we build it into MaxQuant is just straightforward programming. The way we build these things into Perseus is actually relying on the by now multilingual plugin architecture of Perseus. So Perseus is written in C#, and the natural language for programming plugins for Perseus would also be C#. So it has this plugin infrastructure, meaning that you could also write commands for Perseus, and they would then appear as tools in the workflow that you could apply to your data, right? So it's naturally extensible, but so by now, it's not only extensible by C# plugins, but you can also write Python code or R code, or not even write code, but just take existing tools and algorithms, for example, for dimension reduction, and integrate everything you want into the platform. So for example, if you want to use Tisney or UMAP or something like this, you wouldn't have to program that from scratch, or we wouldn't have to program that from scratch. We would just integrate it over these Python and R plugins into the software, basically. So that really opens up for reusage of everything that is developed in the community in this framework. Right, so the first new feature that we developed in particular for single cell data in mind is this so-called isobaric match between runs. So what is this? So this is schematically two LCMS runs with mass going horizontally, retention time going vertically, and then you have these peptide features here, measured and eluting, and so you have actually the same features in the same, because these are similar runs, replicates or something like this. You see the same three features in this run and in this run, right? And what might happen now is that this feature here, this peptide, gets identified because the MSMS spectrum looks good, and so it's then identified as a certain peptide with a certain sequence. And of course, it has these TMT reporter ions here, which are then going to be used for single cell quantification. But then it could be that the same peptide here in another run has not been identified because the MSMS spectrum is of slightly lower quality, and so this might be just above the threshold of being identified. This might be just below the threshold of being identified. And so basically by using this match between runs after feature alignment, so that's actually important that you first do retention time alignment so that you can actually compare retention times between signs. That's of course in MaxQuant already for ages and quite established and working well. So after retention time alignment, also mass recalibration. So mass recalibration is actually also important so that you get very highly accurate masses for these two features. So that just based on the mass and retention time, you infer that this should be the same feature. And because it's the same feature, you can then use the TMT reporters that are in this spectrum here, even though the spectrum itself has not been identified, for quantification, right? So this is basically reducing trying to reduce the missing value problem in single cell proteomics, and it also does, as you will see, substantially. Yes, please.
So in the poor MS2 spectrum that is not enough to support sequence assignment, you still have some peptide fragments that might be informative. Do you use these in any ways for the feature matching? Yeah,
I was actually certain you would ask this. At the moment, no. So we're not. So at the moment, we say, okay, the mass recalibration is so good and the accuracy is so good that we get out of it. Retention time alignment is so efficient and good that at the moment we don't look or try to rescore the remaining fragments here. But of course, we could just build that in as an option exactly as you do it, basically. I mean, it would actually be very interesting to have that in the same platform and just compare it side by side. So that would be very interesting. So. Yes.
Within a run, what about if you had two PSMs? Well, if you had two PSMs from the same elution profile, one of them was filter passing, but one was not. But you had good quant data from both of them.
Mm. Yeah, so that we would actually not recover at the moment. So yeah.
Sorry. I had a question about, so the regular match between run, you kind of align the MS1 data, and then if you have the same M over Z, and you use the MS2 spectra from another run.
Yeah, that's correct.
But here you only require MS-MS spectra, like you do require an MS-MS spectra to be there. So how much improvement do you get here compared to a regular, I would say, match between run, which obviously is not useful for TMT data?
Yeah, so the match between run algorithm itself is actually exactly identical. So it's only looking at the MS1 features also here. So the matching and alignment is not using MS-MS at all. So only after everything is done, then it looks, okay, are there actually other MS-MS spectra that have not been identified, and can I use them, basically? So that's the way it works.
Just to continue that comment, within the same run, if you have several PSMs that match in the peptides, it would be nice to see the statistics of the PSMs, which PSM would take in for the quantitation, because not necessarily the best matched PSM would be a good one. For example, the co-isolation, the abundance, other parameters would be nice to actually incorporate in that decision which one to take for the quantitation, or all of them.
Yeah, so actually at this stage, we just take everything. And then we have additional filters that would select. I mean, you could set a certain requirement at the co-isolation, that one, at least the one that one can see on the MS1 level. One can use all kinds of filters in MaxQuant, but at this stage, we just try to get everything one can get. I mean, there are ideas, I think we discussed this morning, to exactly get these PSMs that are low scoring, but in the same sample, by just allowing them in whenever it is the same top scoring hit as the one that is already identified, right? So that might also help a little bit. But actually there are actually more tools coming that one can throw at it later. So, and we will just integrate all of them, and by that hope to overcome the missing value problem as good as we can.
Sorry, I had a comment about that question also, because that's something that I've been finding, is that the way most software handled the TMT data, the PSM level, that they kind of average, right? Across all PSM they get for like a particular peptide, and that's how they calculate the protein abundance based on that.
That will be the next slide. How we do it actually. Yeah,
but what I found is that if you were to take where the PSM for which you have the maximum intensity, then you get much better quality TMT signal and much more accurate ratios.
That's a very good point, and that will actually come on the next slide. So yes.
While you have this up, so you're showing the example where you have like missing data in one or many runs. But what about when you have data and there's a discordance between the PSM? So maybe there's like, matches between not the primary first ranking PSM, but in each of the different runs, different PSMs are top ranked.
Yeah, so the matching algorithm actually disrupts these matches. So if it sees actually it's trying to combine something that has a different top scoring PSM, it's actually disconnecting these. Yeah. Okay, so now we have more features. More TMT reporters to work with. So what do we do with them then? So I'm only talking about proteins now, not PTM quantification, where you then usually have a lot of PSMs, hopefully, that map to the spectrum, and then the obvious question is how to combine these PSM, these quantities from the different PSMs. And indeed, so one standard procedure is just to take the average or the sum. And that's also the default in MaxQuant, that you would just sum up. And it sounds a bit not the most optimal thing to do, but it's actually not so bad to sum up, because by that you give actually more weight. Actually, you have to sum them up before you, after you really made them like representative of the number of ions. So basically, use fill time and so on to get something that is the proxy for the number of ions. And if you sum that up, that's actually not such a bad thing to do, because by that you weigh the ones that are high abundant and therefore have less likelihood to be contaminated by co-fragmentation, you weight them higher in the sum, right? So that's really something you want to do, right? So if you quantify on the MS1 level, for example with SILAC or something like this, you do something very different, right? Because you're not afraid of co-fragmentation and these things. And in that case, it's actually better to go to ratios, like if you have like a heavy light experiment SILAC, you first go to the ratios within the PSM, and then you medianize these ratios, right? That's the most robust thing you can do. And actually you don't care if it's coming from a low abundant SILAC pair or from a high abundant SILAC pair, because even if you have a low abundant SILAC pair, you're pretty sure that this exists. You know, you saw heavy and light, a few isotopic peaks for both, and you can do decent quantification even if it's low abundant. So that's why it's actually good then to take the ratio of all of the PSMs and then just take the median, no matter how abundant it was, right? So for SILAC, dimethyl, and these things, that's the way to go. That's the best, at least in our hands in MaxQuant, it's the best way to quantify SILAC-labeled proteins. However, that is actually, if you do that to TMT labels, that is a disaster, because by that you just pull up everything that was low abundant and heavily contaminated with co-fragmentation. You pull it up and bring it to the same level of everything that is high abundant, right? So just taking ratios to a reference channel or whatever, and then taking the median is actually not good. That actually is very noisy quantification. So then how can we then use the best of both worlds? So we can still go to the ratios. So if you have a reference quantification channel, then you just take the ratio to that channel. Or if you don't have, then you can just take the ratio to the sum of all channels or something like this, right? So you take whatever ratio is appropriate, and then you take the weighted median of these PSM ratios, right? So it's still median, but so you can calculate median in a weighted way, right? So it still takes the one in the value in the middle, but the single values go in with different weights into this ordered list, basically, right? So what should then be the weights that one is using there? So what we were thinking would be a good weight is actually the number of ions that were selected for fragmentation, right? So in that way, again, a ratio that is based on many ions would have a bigger weight in this median. Something that is just at the noise level and very likely to be contaminated with co-fragmentation signals will get a very low weight in this sum, right? So that seems to be a reasonable thing to do. Just put number of ions selected for fragmentation as a weight, or actually any monotone function of this, right? Who knows that it's optimal to put this just linearly in front of everything. So that's why we in general say, okay, we calculate this number and then we exponentiate it with some as yet unknown exponent, right? So if it's 1, then it's just going in linearly. Otherwise, it's a monotone function. There is a question.
Quite naive question, but with the weights of the number of ions, because you give the trap a certain fill time normally.
Yeah.
So it really depends where in the MS1 peak it was taken actually. Yeah. So this should be actually weighted how the number of ions in the MS1 peak and not so much how many ions were sent for the MS2 to the orbit trap, basically.
Yeah. So actually there are two things one should try. So first of all, if I'm saying it should be, it's basically the MS1 intensity exactly where this MSMS has been taken, not like the apex value or something like that of the peak, right? It's exactly the value where the MS has been taken, and then we want to multiply it with the fill time of the MS2. That should be proportional to the number of ions that goes into the fragmentation, right? Okay, and then, so as I said, it could be any monotone function of this, and so maybe let's restrict the function space a little bit so that we say, okay, we just put an exponent on top of it, which is to be determined. And to determine it in a good way, we first go to some standard bulk data set, bulk proteomics, so that shouldn't really matter that it's bulk proteomics. The thing is that here we have a good control what should come out. So this is a mouse tissue data set from the Kuhn lab with 8 different tissues for biological replicate of mice, and there was also some technical replication. And you would really expect that if the quantification is good, that the clusters really shrink to different organs, basically, right? So there's no one won't really expect that one has overlapping clusters and things like this. So that's really a good data set to monitor and to optimize, for example, this exponent that we don't know, right? So then, we run it through MaxQuant, and so that's what you get here. So this is on the left, the PCA that you get without applying the normalization, and you see a big separation of two clusters, and so the big separation is actually two different measurement batches. So I don't know, 296 well plates, or so this has been measured together here. And then they waited for a couple of days, and then they measured this here, basically, right? So exactly the thing that you don't want to see is basically the biggest separation in the data, and you see that, for example, the tissues are completely mixed, basically, right? But then what you also see is that you have these groups here of 8s, this one here, so that it was 8-plex TMP, so that's what it is, basically. So you have actually two batch effects that you want to get rid of. So one enormous, basically, which is just measurement day, and of course what we were initially wanting to remove is the 8-plex bias that we clearly see in the data. And so then we just apply the normalization that I described, and that's what you get then. So everything is nicely grouping by the organs. So both of the batch effects are actually removed. And so you have to keep in mind this is actually only by applying PSM level normalization. So for example, the normalization algorithm doesn't know what actually the effect is that we're interested in, right? So we didn't specify, okay, these organs should go together or something like this, right? Which actually most of the protein level methods require. So they require that you tell them what is the batch effect that you want to remove, and also what is actually the grouping that you're interested in, right? And then it tries to orthogonalize this somehow, basically, right? So here, even specifying the batch effect would have been already problematic because we have two batch effects, right? So basically, all this information is not given to the algorithm. Still, it basically has the best outcome possible, right? So that means, okay, that's probably the way to do it, and that's also how we want to do it. But then there's still the open question: what is the exponent of the weight? And so we just optimize it. So we look at within group variance, which is, as I said, it's a good thing to look at now because we don't have like overlapping groups that we might have in single cell data or something like this, right? We expect a very clear separation between organs, and that's also what we saw in the PCA of the normalized data that we did. We see a very, very clear separation. So the thing to optimize clearly is just the within group variance, right? And so that's what we do here. So that's the within group variance as a function of the weight exponent, and we see at 0.35, it's minimized. So that seems to be the good value then. And that's what we then also take as a default value, basically. Okay, so that's already integrated into MaxQuant and running. So that's possible to release that actually soon then. So then, yes, please.
PCA controlled by very few components or variables that control this PCA. I can imagine that if some of those entries are very large, the normalization is actually acting only on those components, on those entries, but low abundance ones will not be normalized properly.
So the PCA is only for the display of the results. The PCA is not going into the normalization itself, right? So we only use it here to display the results. So there's no step involved where first one goes into a PCA space and then one would do the normalization there. So I think that's not something to be worried about. I mean, one would then see in the end that there's an outlier or something like this, but it's not affecting like the other parts of the data. Okay. So then we have the normalization algorithm as it is, optimized on bulk data. Now we're going to apply it to single cell data, right? And then we have the two data sets that were already presented here. So this is for the one from the publication with monocytes and macrophages, and Yeah, you know, you have seen the data basically, right? And so then there's the other data set you've also seen that we obtained from Bogdan with the 7 brain regions in there. And okay, so let's see what we get for these. So first of all, what does the isobaric match between runs do with them? So it doesn't look bad. So if you compare just with the number of features with and without match between the new match between runs, it's actually improvement by 102% in the. Okay, these colors are somehow weird. I guess this must be the mouse brain data, and it's 74% increase in the monocyte. No, I think I have it in mind that it was opposite. That I think it must 102% must be no, as I said. Okay, good. So. Well, both are good, basically, right? So that's already helping. So we found that's probably something still to be improved because I think there are examples found that by manual inspection should have been in but weren't. So that's something we're still optimizing at the moment, basically. So, but it can only get better, basically, than that. At the moment, it's missing a few, and we don't really know why. So then what does this look now without and with normalization? So without normalization, so that's the 2 cell type data. It's quite scrambled, and with normalization we see a separation between the 2 cell populations. And that's again with the PCA only, basically. Then, so the question is, so can it be even improved if we put on top of our PSM level normalization some protein level batch removal tools, and then we compared it actually with Limma and Combat, and then also the combination. So this is basically just our PSM level normalization. This is just Limma, just Combat. Then one can also put them together. You can do the normalization on PSM level plus Limma or plus Combat. And you see, I mean, they vary a bit, but they're not dramatically different, right? So it gets a little bit better by putting protein level normalization on top, but it's not a game changer to have it or not to have it, basically, right? So all of them separate the data, basically. Okay, then I guess this is just to show that you can already do t-SNE and UMAP in Perseus, so that's already integrated. So then we, from the brain region data, we did then PCA, t-SNE, and UMAP. And actually, so it looks like, I mean, probably depends a bit what exactly you want from the data. But the PCA separation is actually not too bad, actually, compared to t-SNE and UMAP. Actually, if we did t-SNE, actually these batches were coming back for whatever reason, which we didn't really know. But so I think that's really at the stage that to show you we do all these dimension reduction techniques. I think we even have a spectral clustering already integrated. So they're all there, and like the common user, I mean, maybe you remember we are creating this Perseus tool to have everybody, to give the ability to analyze the data to everybody, basically. That's the idea. So you don't need like a proper education in statistics or something like this. So that's the goal, and these people can now do with the next release exactly these things, basically. Okay, so yeah, so we have a lot of web pages where you will be able to download the software once it's released. So it's not released yet. So the plan, or should I say the ambitious plan, is to release it for the MaxQuant Summer School, which is end of July. But we release it when it's done, I would say, so it's probably better. So let's say somewhere around that or a bit later will probably be release date for the software. We'd also have the manuscript ready then, so that you can just read how it works in the manuscript. And yeah, that's it to that topic. So now I want to get to a completely different topic, which, however, will also be, I think, very helpful for single cell analysis, which is the prediction of MSMS spectra. So that's a more progressed topic. So it was just published 2 weeks ago here. And so the goal here was to be able to predict what an MSMS spectrum looks like just given the sequence of the peptide, right? And then also to find out, so once we can do that, what is actually the benefit for proteomics when we can do that, right? So what is, for example, the benefit for data-dependent analysis, and what is the benefit for data-independent analysis that one can get out of such a tool? Right, so as I said, so the problem setting is very, very simple. Somebody pulls out a sequence out of the pocket and then says, okay, let's say I use this fragmentation method, this mass analyzer, and so on, these kind of properties of the system. What is the MSMS spectrum going to look like? So that's the task, and so the team is actually, so we're collaborating with the Very Life Sciences, with Peter Zimmermanzik on this side and his team, and on my side there are three graduate students sharing the work on this project. So what are we going to use? So I should actually mention that there was actually another paper back to back in the same issue, working on the same thing, and so actually they are actually located north of Munich, and we are located south of Munich. So everything is somehow centered around Munich, and I mean, local people know that the south is actually much nicer than the north, but so you can ask everybody, I think. And so what are we using as a data set? So that's actually a slight different to what they were using. So because they were doing all of this with synthesized peptides, so we don't. We just use Pride, basically, right? So we take a big chunk of data out of Pride, reanalyze the data with 1% FDR, whatever FDR setting, and just take that. And so in our opinion, that should actually be good enough, because so if the machine learning algorithm is really that vulnerable to false examples, like then it's probably not going to be useful. Or, I mean, I don't know what autonomous driving will be if that would be the case, right? So, I think that's really not necessary to make a library of synthetic peptides. You can just take Pride and that's it, basically, right? So that's what we did. We took 25 data sets spanning 5 organisms, resulting in 60 million PSMs. And so they are then split into 90%, 5%, 5%, training, test, and validation data sets. And you see here what, I mean, the typical coverage of properties over chart states, fragmentation methods, 5 species, and so on and so forth. And then we use that as a training data, and we use then TensorFlow as basically the engine for making the prediction on this data set. And so the model that we use, the architecture, is a bidirectional recurrent neural network, which is probably most known for natural language processing. So the nice feature of this kind of network is that it takes a variable length input here. So it has to. So if it's actually used for natural language processing, then every word, of course, has a different length. So something that would work with a fixed length representation, like linear regression or something like this, is not very useful for natural language processing, but also not for analyzing peptides, because as we know, peptides also come in different lengths, right? So fixed length representation is not a good thing for learning peptide properties. So then it has this input here of variable length, and what comes out here is basically then a fixed length representation that is then much easier to handle. And here you can now also fit in metadata. So here you can tell the machine that, so I was actually using HCD to fragment this peptide, or it was this and that charge state and things like this, right? So here feed in all kind of metadata, or the fragmentation energy that was applied, right? And so the idea is that you have this one model, so there's only one model that knows everything about peptides, right? So you don't need to retrain models for all kinds of different classes of peptides, not even speaking of length of peptides, which would be crazy, basically, so that we all don't need to do. And then it ejects then on top the intensities for the ions that you're interested in. So B and Y ions, but also, for example, neutral losses of water and ammonia and these kind of things. Right, so then we have our data, we have our model. So how good is the model then? So here you can see an example where it could actually predict the spectrum quite well. So the reason why in the predicted spectrum you don't see a peak for every experimental spectrum is, of course, co-fragmentation again, right? So it's only going to predict the peaks from the peptide you're interested in or that you thought you were isolating. It's not predicting co-fragmentation, of course. But then, yeah, so it can predict one spectrum, but I guess every algorithm can predict one spectrum. So how is it on the whole population? So on the whole population, it's not so bad. So this is, these are distributions, so the box plots are distributions of correlation coefficients between experimental and predicted spectrum. And we have this here for, so deep Mars prism, that's what we call the model that knows everything. We have the same for like the best conventional method, MS2PIP at the time. And we have it here as the theoretical limit, because the limit is actually the correlation that you get between technical replicates. So if you just measure the same spectrum twice, of course, it will not be the same. So there will also be some deviation. And so that's actually the distribution of correlation coefficients for these technical replicates. And that's kind of the limit, that's a theoretical limit for the performance of the prediction. And that's we're actually getting very close to. So we are actually almost reaching the theoretical limit for the prediction quality, and by far outperforming conventional methods for doing this. And so that's actually true for all kinds of slices. You can now look into the data. So if you look for different charges, so you get for each charge you get the same pattern. And that was actually, so for example, charge 3 was predicted very badly before with these methods, and so that's actually not the case anymore. Over all the length bins, mass analyzers, fragmentation types, you see the same order of these three distributions. But then we made another model as well. So why would we do that? So we already have a model that's like unbeatable, basically. So why would we come up with another model? Well, because so maybe we can have a simpler model that is much faster to retrain, because actually training the deep Mars at the moment with the 25 data sets already takes a couple of days on Google Cloud. And you can imagine if we integrate more and more data from Pride, that will take longer and longer, or more resources, however you parallelize it. So it might also be useful to have a model that is maybe a little bit worse in prediction accuracy, but instantly retrainable, right? So that obvious application would be, it could be just sitting up front MaxQuant, and whatever unexpected data you have, like single cell with TMT and crazy carrier channels and so on, which might be quite different in nature. The MSMS spectra might look quite different than what we have sampled in Pride, right? But then this model could just be trained on the fly, and it could be used in MaxQuant basically to improve the spectral detection, for example. So this other approach is a sliding window-based approach, and it's not so different from other sliding window approaches where you try to predict secondary structure or solvent accessibility. So that has been done for, I don't know, 100 years or something like this, at least feels like. And so we do the same here, just on the peptides, and basically always center the window around the bond for which we want to predict the intensity, right? And then we move the window along the peptide and predict intensity one by one, basically, right? And so this can then be put into a fixed length representation. So we just use a fully connected two hidden layer neural network to do the prediction, to do the regression on these. And so what do we get then? So in terms of performance, so that's the things that we saw before. This is now the new method, and we call it winner, even though it's not really winning. But so it's actually window-based neural network that is easily retrainable. So that's why it's winner, basically. And so you see, it's still better than MS2PIP. It's not quite as good as Deep Mars Prism. But it's okay. It's doing well, right? So it's better than the state of the art, not as good as the best. So yeah. Okay, so we can actually do exactly what I said. We can use this for situations where we want to train, like maybe in half an hour and not in like a week or something like this, right? Like in the time a MaxQuant run takes, basically. Okay, good. Yeah, so one other interesting observation is that this deep learning model is actually highly interpretable. So that goes probably against what you hear all the time, that so these are very good at making decisions, but nobody knows really why they're and maybe they're going to kill us all or something like this, right? So that's I think that's what people think and what's in the newspapers all the time, but that's actually not true. So there are actually new methods. So this one called integrated gradients that just came out of Google that help you to interpret the model. So, for example, and this is actually instance by instance. So it's not like for the whole training data, it's telling you what feature is important, but for one instance, it's telling you what was actually the important feature. So you would get matrices like this here, which tells you for predicting this particular intensity here at this position, which of the other bonds, which of the other positions in the peptide were actually important to get to this decision, basically, right? And then if you make some statistics over many instances, you see actually interesting things like, first of all, differences between the determination of Y and B ions. So this is B on this side, Y on this side. So they look quite different in their distance characteristics for which side is responsible for the intensity at which bond. And also it's quite asymmetric, right? So it's actually quite a difference if you go to the N terminus or to the C terminus. So probably all things that are not too surprising. Also that you have long range interactions is not too surprising, because otherwise it would be a simple problem. You would just have to look at the two neighbors and you would be done, but we already figured out that's not the case. So that there are long distance interactions is not going to be surprising, basically. So one can interpret the model and also recover lots of things one knows already. But I think, I mean, we're just touching the surface. So I think I'm very glad if other people examine the model and try to find interesting structural things in there. Okay, then, so what can we, so now we can do these good predictions. What can we do with it? So what can we do, for example, to improve DIA data analysis? And so then we ask the question, so is it actually necessary to measure these libraries at all, right? So one can do this in several levels. So first of all, one could say, okay, I know which proteins are going to be in the library. I know which peptides are going to be in the library. Maybe I even know their retention time, but do I need to measure what the spectrum looks like, right? And so to investigate this, we just erase the spectra from a library. So it's a plasma sample with a plasma library. Erase the spectra, replace it with the predictions, and that's basically then what you get here. So the amount of quantified peptides is near more or less the same as what you get with the original library. So that's original library. This is replacing spectra with predicted spectra. So at least the fragment intensities you don't need, basically. So what about retention time? So then we also replace retention time. This is prism plus drip here. So now also retention time is predicted, and you see a little drop, but it's still almost as good as using the original library. So also retention times you don't need. So actually the only thing you need the library for is to know which proteins and which peptides are in the sample, right? So that's of course not an obvious thing to know. And also we compared, of course, again against Leonard Martin's, which is not as good at all, basically, so as our methods. So yeah, so one can now do many things, of course, with this, right? So you could, for example, if you have a library already measured, and you say, yeah, but on top of that, I would really like to look for a few other peptides, like for example, so this is from a human sample, and there might be just a few SNPs in there, right, which you don't measure. Which you didn't see when you did the library, right? And you could just add them, right? So you just mutate your peptide, get the prediction, add it to the library, basically, right? Or, I mean, one can also be more ambitious, of course, and try to predict the whole library, maybe starting from RNA-seq and observability or flyability predictions of peptides for these proteins that you would expect based on the RNA-seq. But that's still an open question if that's really going to work or not, right? But that's definitely something that we're going to explore. So looks like it will benefit DIA data analysis a lot. So how about DDA? So we have this Andromeda search engine integrated into MaxQuant, and that's actually where it gets interesting for single-cell data analysis. So can it improve identification rates and things like this? Then it would be a cheap way just to get higher coverage in single-cell data, basically, right? And so it can. And the way we use it in the Andromeda search engine is that so how do we now make use of the intensity? So we already did make use of intensities to some extent in Andromeda by just scoring it better if a high-intense measured fragment is explained. So if a high-intense measured fragment is explained, that's better than if a low-intense measured fragment is explained, right? But it was asymmetric, so that was not the same in the other direction. So the theoretical spectra, nobody cares if like a high or low intensity fragment is explained. But now we can make it symmetric because we know now for each sequence that we offer, for each peptide that we offer, we know what the spectrum should look like, and we can make it completely symmetric, right? So it's now also better if a theoretical fragment that is supposed to be high intense is explained than one that is low intense, right? So one just makes it symmetric. And so what do we get? So first of all, this is a comparison on a set of synthetic peptides from north of Munich, where we just plot the old Andromeda score against the new Andromeda score. And so you see two things. So in the range of high scores, it's not changing. And that's a good thing, because when the score was high, we were claiming this was a good identification, right? So if this is now changing, that would be a disaster, right? So it's not changing, that's good. But then for lower scores, the score is actually getting bigger. It's always getting bigger or staying the same, but that's by construction, because the way the score is calculated is actually calculating many scores over different numbers of top scoring theoretical ions. And one of the things it's optimizing over is actually the old score. So it can never get worse than that, basically, right? So it's getting better for some. I mean, here you just see again, that's a histogram of the score differences between the two versions. So many are staying the same, but then you have this tail of increasing score. And so on the synthetic data set, we actually know the ground truth then, and we know so I can just show you some examples where it's basically correcting the wrong decision it would have done with 1.0. Like here, where it's actually getting the right version of two neighboring amino acids that are flipped in the old score and that are in the correct order with the new score, right? So the EIVVI flip basically, and then but there are of course other examples where it's just a completely different peptides, like in this case here. They don't have anything to do with each other, but it goes in the right direction. The new score is actually top scoring the correct version. And it's correct because it's synthetic peptides, so that's why we know it's correct. So then how does it look if we evaluate it on a bit more complex data, like for example, HeLa data set, which you can see here. So you see the same trend, the high scores are not changing, the low scores are staying the same or getting bigger. And then if we make the evaluation, so how much does it actually help in terms of gain in identifications? So that's what you see here. So the gain in PSMs is Q value specific, and so if you're in the very high specific range, it can go up to 20% on the 20 percent points on this HeLa data set. And so one should actually say that's actually not a small number, because so this is like very standard HeLa data set that's very well characterized by MaxQuant. So the identification rates are already high, and also many of the non-identifications are actually modified peptides and things like this, right? So that's actually a good gain. So if you just get that standardly with every HeLa run you do, that's nothing you would want to miss, basically, right? And so my expectation is actually if you go into more difficult situations, and that's actually also something that the people from north of Munich are showing in their paper. So if you go into a more difficult situation like proteogenomics or something like this, the gain factor is much bigger, right? So if it's more challenging, it's helping more. And so I'm sure we have challenging data in single-cell proteomics. So from that, I would predict that it will also help a lot there, right? right? But that remains to be seen. And so we did it now in a proof of concept study. So in this form, the machine learning was not 100% integrated into MaxQuant, but it was actually working in some sort of batch mode where it would write out the PSMs that it needs the information for. Then we do the training outside of MaxQuant and feed it into MaxQuant again. But so that's of course not how it's going to stay. So we're working now and pretty far in integrating that seamlessly into MaxQuant so that on the fly it's going to make its predictions and use them in the scoring. And once we have that finished, I think the first thing we should do to apply this exactly to single-cell proteomics data, where it should then reduce the missing value problem. Okay, so that's the two stories that I had. So acknowledgements. So Sung-Wan, of course, who was already speaking 2 days ago, did most of the single-cell work, and then Petra and Shivani and Fabio are the ones responsible, at least for my group, for the spectrum prediction. And thanks a lot for Nikolai Lab and Bogdan Lab for always helping us. And thank you for your attention.
Thank you. I can see Marco has a question.
So for the deep mass, which is really great, but I guess it depends a lot on the collision energy and the calibration of the instrument. Can you put this a little bit as an input first, that you have a normal mass-spec run on your collision energies, you ram through and then put this in as a deep mass so that you then correct automatically for whatever you predict? Or
yeah, absolutely. So I mean that's one of the. I mean there are a lot of next things to do which are like obvious to do, and they don't require thinking. They just need to be done. So one is that we feed through the collision energy. I mean the other thing that's also so at the moment we are only doing predictions for unmodified peptides and methionine oxidation. But that's easy to go beyond that by just declaring the modified amino acids as 21st, 22nd, and so on amino acids. So at the moment, it's also heavily biased toward tryptic peptides because that's what you find in Pride. So it just needs to see non-tryptic peptides, but there's no conceptual thing one needs to do, basically, right? Calibration is not necessary because, that's MaxQuant taking care of, basically, right? So, you can work with uncalibrated instruments.
But I guess the fragmentation really depends a lot on the collision energy and your settings, basically. So, but it basically, MaxQuant would read it out automatically?
Yeah, it would read it out automatically.
And then readjust deep mass based on this?
Yeah, exactly. Well, it would just need to feed it in as one of these metadata parameters.
Okay. And I guess because you mentioned modified phosphorylation, I guess the learning data set is not so big there. So that might be a bit of a problem because you don't have 1.4 million unique peptides, but probably a much lower number.
Yeah, phosphorylation is not so bad, I would say, but if you go to other PTMs, then of course that might become an issue. But then, the way to go might be also to use Winner, because Winner, being like conventional machine learning, needs less training examples.
I'm sure the south of Munich is much lovelier than the north, but aren't you concerned that the learner algorithm is just rediscovering the biases of MaxQuant as opposed to the approach using the synthetic peptides where you don't have a cutoff for matches? And can you extend it beyond MaxQuant?
Well, not necessarily MaxQuant, but the peptides that we identify, basically, right? So, yeah, sure. Well, we know that it generalizes because we have a test set that has not been seen by the data. So, if we would do severe overfitting on the data that we have and would only be able to make predictions on the data that has been seen, then actually the performance should not be as good because we really use independent data to assess the model. So I'm pretty confident that it's not learning the biases of MaxQuant. It might be learning the biases of tryptic peptides, but not the biases of what we can see with MaxQuant.
So I have a couple of suggestions. I really want to generate strong data to benchmark the improvements that you're introducing. For one thing, it is clear that the PSM normalization will benefit if there are multiple PSMs per peptide or per protein. And currently, we don't try to do that because we want to have high throughput. It is very easy for us to increase them by a factor of 10, and then we can potentially see much bigger benefit to evaluate how well it's performing. The same with benchmarking the removal of the batch effect. Instead of just looking at separation of cells that may have genuine biological reasons not to separate, we can already give you a lot of data from single-cell lysates, from lysate diluted to single-cell level, where we have very strong expectation that we should always be measuring the same, and then we can rigorously benchmark exactly how much is error and how much is improvement. So there are some really simple experiments for us that we can do to provide stronger benchmarks.
It's great to hear.
Thank you again, Jürgen.